Thank you, everyone, for coming. We have a lot of ground to cover in a half hour. We'll get to some questions at the very end, so if you can be thinking of them, writing them down. There will be 5 minutes, so they'll have to be really quick, but we'll get to some. I'm going to moderate a discussion here about the dawn of this new era of medicine. So I figured we should start a little bit at the top. I know you guys probably know most of this already. It's a pretty savvy crowd, but like we were saying backstage, Ed, a lot of people don't even know what precision medicine is, and you have done a lot of work as an advocate for public communication among scientists. So how do you concisely tell people the backstory, what it is, and the potential of it, without rambling on so long that people
I'll try to hit the nail on the head and be very quick.
How do you do it in conversation, I guess, because it's a tool a lot of us could use.
Yes. So essentially, personalized medicine or precision medicine is based upon the assumption that people are different. And obviously, we've known that for a long time. But in the last 10 or 15 years, we've developed tools that allow us more precisely to understand the molecular differences among individuals. And that has, in turn, allowed us to link therapies to diagnostics, which also, in turn, produces better outcomes. And we would also argue that through personalized medicine, by making it more efficient, the health system will also benefit. So this is essentially a good thing. It gives modern medicine what medicine has wanted going back to Hippocrates. That is to be able to treat individuals according to what's going to work best for them. How's that?
Yeah, I mean, it sounds inspiring, uplifting. I should say that I am moderating at the end an ethics session, so like we're not going to delve into any the concern side of things. I wanted to. We'll talk here more about like just like you said.
So we'll be
what's
on
the promise?
upside.
Yeah. What is the upside here? What is the dream? And then we'll work backwards to kind of some specific things that you guys are doing to see it happen in the short term.
Can I ask Ed a question?
Yeah, please.
Oh no.
We just met. So you're doing communication to the public, but what's your thoughts in terms of educating physicians who have to actually deal with this massive amount of data?
Well, actually, that's a great question. And we actually have surveyed physicians, and it turns out that only 10% feel they have enough genetic information in order to treat patients appropriately using genetic or genomic information. So we know that among providers, there's a lot of work to be done in terms of education. And obviously that's even more true of the public, which is in part why my coalition was created, to be an education and advocacy organization. And I would also say along those lines, and this is really a very important point, we don't get to choose the biology we have to deal with, either as physicians or patients. We just have to deal with it. And so we have to adapt our systems in order to deal with it, and that includes the entire spectrum of healthcare. We just can't ignore biology. It's a given.
So you're saying that doctors don't have enough information in terms of their own education around genetics?
Most physicians were educated before the human genome was mapped.
Right.
The overwhelming majority. So most of them don't know what a particular mutation is that is going to determine the success or failure of a treatment.
Right. So the pace of technological advancement in the field, maybe you could talk a little bit about that, Dr. Nelson. And it sort of gives us an idea that these things we're talking about in the future are not so outlandish when you see how far we've come in the last 2 decades.
So I joined Craig Mentor's Institute about 21 years ago this year. And back then it took like 2 years to do a microbial genome, and the human genome was this massive long-term project. And now we're doing a genome probably in a couple of days and a microbial genome in a couple of hours, and it's just costs have changed significantly. Technologies have improved so much in the past 20 years. I agree with you that most of the physicians were never really exposed to that. That's why we need to have training. So Oh, you can't hear me?
It's like every time you turn to the right, it becomes very hard to hear.
I'll just look at you then. So I'm just saying that technologies have changed so much in the past 20 years from where I sit that I can only imagine that it's going to continue to advance and improve, and there's going to be massive amounts of genetic information for everybody to work with. I mean, Claire mentioned this concept of big data, but it really is a lot of information that's going to be available for an individual to play with. I imagine that people are going to have their own genomes in their pocket at some point.
Yeah, and so that's what Aris is working on, making that data pool huge. Your company is Regeneron,
Yes.
And you have sequenced 200,000 genomes?
Close to, approaching that, yes.
Close to?
So this is the huge opportunity and the huge challenge. So I 100% agree with both of you guys and what you said. Personalized medicine is nothing new. We've always been trying to get the right therapies to the right patients. What we do in terms of understanding disease, making drugs, treating patients is one of the hardest things we do in society. And building a foundation in the genetic code, understanding the molecular basis is going to be key to doing that. And the tools are amazing. There was a really nice article, I think Matt Herper wrote about what he called Flatley's Law. And the innovation and the pace of technology, what Karen was talking about with sequencing, has far outstripped Moore's Law, what was kind of famous in terms of electronics and semiconductor industry. And I think we're at that point where we're having that kind of semiconductor moment in healthcare, where genomics, we're sequencing hundreds of thousands, millions of people, and it's now becoming more ubiquitous, routine in medicine. The challenge, though, is with all this data, how do we interpret it? And we have so much information, and we haven't yet linked all of it to causes and understanding the correlation with disease. And we have this, it's not clear-cut all the time. There are some very clear-cut examples where these mutations lead to this disease or these patients respond better, but there's so much information out there that's in this kind of indeterminate range or unsignificant findings. And I think that's going to be the challenge. How do we get physicians up to speed and providers in really understanding all this and making it actionable?
Well, can you give us one of those clear-cut examples of where something seems to be showing promise, that we're actually doing something with this genomic data that's already changing clinical medicine?
Absolutely. So I can give you a very close-to-home example. Our work with the Geisinger Health System, we've sequenced almost 100,000 individuals in that health system. And there are genes and mutations that most know about in clinical care. When you think about things like familial hypercholesterolemia or cancer predisposition, BRCA1 and 2, recently made very topical, Angelina Jolie, those types of stories. And it turns out if you take an agnostic look at sequencing and a population, a healthcare system like Geisinger, it turns out that about 4% of individuals, that's a lot, have these incidental findings, things that a health system like Geisinger feels they should act on and return to these patients. And there are amazing stories, participants who've joined this study. One woman, Barbara Barnes, a patient who's talked about this very public. She's a grandmother taking care of 3 kids. The parents are not there, and she's solely responsible for them. And she found out she had a BRCA mutation, was pretty much asymptomatic, did all the screening and came out negative, but decided to get her ovaries removed and fallopian tubes. Turned out she had a stage 1 cancer. Very good prognosis that she found it then, but as she says, if she wasn't in the study, didn't know about this type of information, and had waited till she was symptomatic, it would have been a very different story. I mean, she says this type of stuff saves her life. So those are clear-cut examples, and there are many of these. And there are other things like in drug development and in medicine, we look at markers, like these immuno-oncology markers for these new medicines coming through that really help look at who might respond or not. But there's a sea of things far short of that where you look at it and you say, we just don't know. I mean, is that causal? Is it really going to change a patient's course?
Right. Okay, both of you at the same time.
Ladies first.
No, sorry. Yeah.
Sure.
So just following on that point, is Regeneron now developing drugs for targeted populations, subpopulations, as opposed to one-size-fits-all, trial and error?
So we've always been trying to do that, obviously. And sometimes it just turns out that, we've been really a genomics-guided drug discovery and development company, whether it's been mouse genetics and now, with the tools, human genetics. And so I can point to some of our early drug approvals, and these were drugs that directly followed a human genetics insight and a discovery. So they were targeting exactly kind of that genetic lesion that's causing disease. So in that sense, it's targeted and one size fits all because it's for that unique population. But yes, for other drugs that have broader indications that we're moving forward, we are constantly looking at interesting subpopulations defined by genetics or other molecular markers. And one thing I think is now becoming commonplace in the industry, and it's our practice, is we are sequencing all samples from our clinical trials. We are looking at all substrata based on various pharmacogenetic markers and trying to see if there is anything there that's worth maybe stratifying, doing a prospective study, and seeing if really they should be even more specific to certain populations.
I have 2 quick questions.
Okay, yeah.
Do you go back and look at like failed trials at all? Like the data coming out of that? That's question 1. And just for clarity, you're doing exomes, right? Not complete genomes, because there are other organizations that are actually doing complete genomes. But you mentioned earlier on that you're focused on just looking at the exome, and I was just wondering if you could.
So we're just in the entire genome. We look at our approach is this is kind of getting into nuances and technical details, but we absolutely focus on exome sequencing and then genotyping,
right.
Which these days with imputation, all that kind of stuff gets you amazing coverage across the entire genome. So essentially, what we're focused on is really large effect genetics. That's the key, right? So we're not looking at things that are marginal or on the fringe.
But there's a lot of potential there, I can imagine.
There's a lot of potential,
yeah.
But we just don't know. And the genes obviously are the key to the genome and what regulates them. And so I think it's been well documented that really the genes are where the largest effects in the genome come from, and that's where we focus.
So how does the industry shift from right now wanting to produce something that has a large market to something that has a market of one person? Why would anyone ever do that? I mean, I'm not a business person, but I watch Shark Tank, and they would be like, how big is your market?
Well, we don't think
one
like that.
person.
We don't think like that at all at Regeneron, and I hope that the industry doesn't think like that. It is not about the market, and we can cite so many examples. One of my personal mentors, I'm a clinician by training as well and a scientist background. I was at Duke, and my immunology mentor, Tom Tedder, was involved in the cloning and discovering of CD20, which led with Ron Levy to Rituxan and Biogen and Roche and all that kind of stuff. And the story of that drug, having gone through so many different companies, is because each company looked at the market potential, not the science and the impact on diseases, and kept tossing it around, didn't think it was right, and it was one of the most successful drugs ever. And that's happened to us at Regeneron over 30 years. We can point to several of our products where people didn't think it was a big market internally, or some of our partners, and we persisted. You really can't think about the market. It's just speculative. It's guessing it at best. You really need to focus on where's the best science, where's the biggest clinical unmet need, where can we make an impact on patients. And if, like Roy Vagelos, our chairman, and our entire company believes, you do well by doing good, and the profits and all that kind of stuff will follow. And that's absolutely been our mantra, and is really the only way.
This is what I meant. You have to pay attention to the science.
Yes.
The science tells us that people are different, so in order to develop drugs in the future, we're going to have to develop them more or less for subpopulations. And once upon a time, the pharmaceutical industry may not have been there, but now they realize they have no choice. And they are there.
Does that mean drugs are just going to cost a ton of money? I know that if every article that we publish on The Atlantic was unique for every reader,
but we're going to be
a higher cost of subscription.
We're going to be in the future paying for value and not necessarily for volume, whereas that may have been the case in the past. So if we're paying for value and people are getting better, they're living longer, and or getting cured, society is going to benefit, and there will be downstream benefits to the health system, which won't be proposing inefficient and unhelpful treatments, which are also expensive.
Okay. Imagine
the money you could save by not giving medicines to people for whom they don't work.
And we're not all driven by the market and the profits. I would just say that our companies, we have large dedicated workforces of scientists and clinicians and lots of other folks who are committed to making a difference. There's plenty of opportunity for financial gain and all that kind of stuff, but there's a lot more in motivation. And all of our companies have opportunities where they're probably zero, big commercial opportunities, whether it's vaccines for emerging world public health opportunities, our involvement with various antibodies for infectious diseases. There are many chronicled stories where there was really zero financial gain, but the motivation and the reward to the scientists and the clinicians and the companies, you can't underestimate how important that is to an innovative company and an environment of making game-changing breakthroughs and bringing new medicines forward. It is an essential part of it.
I don't underestimate that. I mean, it's just about having a viable business. Where, you have to. And I'm overthinking this. I'm talking about a drug that's going to one person. Yeah. We're talking in the interim about not giving every single person the same statin, what's a realistic timeline for having drugs that are just? How many more new drugs are we going to be seeing, and how much smaller does the market get per drug in the next couple of years?
Part of the opportunity here is it's faster time for drug development, and the other thing is that if you can identify the subpopulation for whom it works, the effect size is demonstrated more easily. So, for example, Pfizer developed a drug called crizotinib, which only works for 4% of patients with non-small cell lung cancer. Because they have a particular mutation in the ALK gene. Amazing results from this drug, but it only works for 4% of the patients.
Yeah.
And that example can be multiplied many times. And this is where the future of the industry is, and this is where the future of hope for patients is.
Okay.
So it doesn't get down to the single patient, but it does get down to
subpopulations.
The global picture, we all have to deal with is we talk about the 90% failure rates. If you aggregate all those costs, we're spending billions of dollars per drug approved. But what Ed is talking about is that math changes when all of a sudden you incorporate more effective strategies, whether it's genetics or other ways of getting precision medicine, where the success rates get better, and it's cheaper and easier, more effective to do drug development, and therefore, in terms of trying to recoup those costs with pricing, you can do better in terms of pricing for value.
Okay. Can we take a kind of radical swing?
I can't answer any drug questions, so.
That's fine. And but there's nothing else, I think, this morning where we're talking about anything other than the human genome. And you've done a lot of work with the microbiome, which it's going to seem like having a sequenced microbiome, knowing what kind of microbes we're dealing with, is going to affect these therapeutics too.
Yeah, I would think so. So,
yeah, where is that going to play in? I got my microbiome sequenced.
Oh, great! I can ask you about that. That's awesome. So,
I didn't learn a lot.
I know you didn't. So that's a great question. So I actually did the first human microbiome study in 2006, and for me, it's just amazing to see. Can you hear me? Yeah, how it's taken off on a global scale. I think there's significant potential, but I really feel like it's the early days yet, because there are people like you who got their microbiome sequenced. And
I did it for journalism.
For journalism, awesome. But what did you get? Like a report that said you have
I might
two different
be
kinds
obese and depressed.
You might be obese.
Well, we know you're not obese.
Yeah, yeah. We did some introspection after that.
Yeah.
No, but it was like correlating, certain.
Right, but you're a physician and you don't
like,
really understand what it means, right?
Yeah.
And it's a single time point, and it probably depended on what you had for breakfast that morning.
Yeah.
So I think as a community, we're really in the early stages. A lot of the studies that have been done have been on cohorts of 30 to 100 people. You're trying to extrapolate to the rest of the globe, and I really think a lot of work remains to be done. But I do feel that there's significant potential there. I think in terms of developing new diagnostics and therapeutics, we definitely will learn from the microbiome. And, you can look at healthy people who live to 105, and they might have unique species that can benefit the rest of us. So significant potential there. There's some correlations with some diseases that seem pretty robust. But in general, a lot of people are like you. I got it sequenced and not quite sure what it means.
So what this company Ubiome is doing is sort of acquiring then tons of data from everyone who pays to get it sequenced, and then if you can correlate that with lifestyle and medical records and whatnot, you can eventually start to tell people. But there's this constant race in human microbiome sequencing, in human genome sequencing, in microbiome sequencing, where you need to buy in, you need people to give the data, and then also you don't know exactly what to tell them to do with it yet. So how do we,
Right.
we've got 2 minutes left, how do we navigate that responsibly?
I think
And
that,
then we'll go to questions.
okay, so I think that there are a couple of companies out there, and I'll put a plug for human longevity, where we're actually sequencing everything that's there. Some of the companies you're talking about are just looking at a single phylogenetic marker, which is not telling you about everything that's in your system. And I think we need, as a community, to move away from that and really try and correlate the microbiome with the genome and look at those kinds of correlations. And I think that that's where the future is headed. I
Just
don't
get
know.
as much data as we can.
Well, I don't know if we need, I think we need reliable,
a
clean
lot
data.
of reliable data.
Reliable.
Really hard data.
Yeah.
Right. And don't try to make too much of it at the beginning.
You're not obese.
No.
No.
I might have a propensity for it.
Yeah, but I
Play
don't
it
think,
safe.
I could have told you that without you paying for that.
Yeah. Yeah, well, so that's why, the physician's clinical exam is still important.
But I think it would be great for physicians to become aware of the microbiome concept,
Are they
because
not?
a lot of people are really excited about having stool sequenced. I have no idea why. I get the most bizarre phone calls. And I think that's another, the physicians need to be able to have this conversation with their patients about what it could mean or could not mean.
Oh, yeah. So you tell people that it's worth doing right now, other than to contribute to the greater good of our later knowledge?
If they're going to do it, I suggest that they do the complete metagenomic sequence and try and see if they can integrate it with their host genome and look at relationships that way.
Okay.
Something will happen.
Okay.
But I think it's very much a research effort. I mean, I think the question was, is really not much that I'm aware of that's kind of clinically actionable.
Correct.
So
Right.
That's absolutely correct.
You're sort of paying for the opportunity to contribute to research, which is cool if you can get people to do that. Can we go to questions? We have 5 minutes. Yes.
Hi, I just have a question, actually from the patient consumer standpoint. What are the options to actually getting your genomes, the gene mapping done? Because most doctors don't talk to patients about this at all.
There's several groups out there where you can get your, there are several groups out there where you can actually get your genome and your microbiome sequenced. I think the concern is what you do with the data. If your physician doesn't have exposure to genomics, like what's going to happen there. So that's the conversation. But there are places that you can get your genome.
It's like 23andMe is the famous.
Human
So
longevities is another
Right.
place where you can go and get your genome and your microbiome.
there are times when
Completed.
it will be clinically indicated, and that's
Yeah.
a small fraction of the time. And then from a direct-to-consumer standpoint, 23andMe was mentioned. Human Longevity, I think, is doing these kind of
does it, yeah.
executive genomes as well. But the challenge is, what do you report back, especially when you're not in a healthcare kind of provider setting? Like we all just talked about, it is very hard to interpret and understand the consequences of what this information means. So I think that's a challenge.
Yeah, we'll talk about that a little bit more in the ethics session too, because there are a lot of
The
things
issue is
people
clinical utility.
clinical utility.
But some of
Is
it
this
is.
is this a good use of
especially if
resources?
you're going to sequence a cancer tumor to figure out what's likely to respond.
Right, but a lot of it is actionable, but you probably
Yeah.
need a physician who can help you interpret what it means.
Yeah, that's kind of the argument with like direct-to-consumer marketing and
Yeah.
23andMe, and it seems like most physicians are saying that's maybe best to be done in collaboration with a physician rather than just choosing to do it on your own.
But as you were saying earlier, most physicians were trained prior to this, so that they
Yeah.
really don't. Do they see the value? Would they even suggest it? I mean, so how does that? You're sort of caught as a patient.
Well, it's an educative process. It depends if you're dealing with a sick patient or you're dealing with a well patient who gets tested by 23andMe. So there's a whole spectrum of things. And this is an emerging field, an emerging science. And its clinical utility is still being determined. So, I mean, that's the exciting thing about what we're talking about. We're really on the frontier of science and medicine.
Okay, another question back there.
Hi, thank you. Amy Raskin from Chevy Chase Trust. I have 3 questions, a 3-part question.
Could we do just one of them?
We've only got 2.5 minutes.
Yeah.
Okay, well, I'll throw them out there, and if we choose to answer whichever is easiest. But you said 4% of the people have actionable results right now from getting their genome sequenced. What could that number be? I mean, we recently saw the Novartis test for the Alzheimer gene related to Alzheimer's. Do you think that number is going to be significantly higher in the future, where we'll have more actionable results? And then there was an earlier question about Watson. Are you using Watson to analyze your data? And then my final first question was, do you view the NovaSeq, or for everybody, do you view the NovaSeq platform as a game changer and an accelerator?
Okay, any of those strike a particular chord?
Yes to the last. Yes. No to Watson.
Same on those two. And then on the first one, absolutely.
Yeah.
It has to increase. So,
Do you
right.
have an idea of what it could be?
I have no idea. But the 4% number is actually things that are really actionable. So there's way more than that that is meaningful. But if you're going to actually work with the patient and change maybe their course of their care, those things are, for instance, 76 genes that Geisinger Health System selected that they are very confident in, that they feel they need to return to patients, and that they would do something with their patients about their care.
That was remarkably concise for 3 questions. Can we get one more question?
Let's get 3 more.
Hi, I'm Ken Habarda from Sparks and Honey. Ed, you mentioned in the beginning that we're kind of stuck with the biology that we're given. I feel like with the speed of technology and, again, the speed of access that we have, in the previous talk, she talked about the ability to kind of change our sequenced DNA, which would be like designer babies, is kind of where I'm heading towards. And as that kind of technology speeds beyond our comprehension, as a layperson, where do you see like CRISPR and the ability of having that fall into just people's hands?
The technology you're referring to, it gets to gene editing and ability to change our genetic inheritance, and that's pretty powerful.
And you have 10 seconds.
But in 9 seconds, I just want to clarify that when we're given a biology, I mean, human biology is something all of us have to deal with, not at an individual level, but collectively when we develop drugs or put in place reimbursement systems or regulatory systems. We have to understand that the principle of human heterogeneity is there to stay.
We will be talking more about that in a later session about editing and the ethics surrounding that and questions about using it responsibly. Thank you so much to the panelists.